Hey everybody, I think this is going to be our last video this semester on translation, at least the mechanism of translation. Before we start, before we really get into translation here, we need to discuss the structure of the ribosomes, which are the main things that perform translation. So we will focus on ribosomes in eukaryotes. And the ribosomes in eukaryotes and in prokaryotes are very similar. Essentially, the components, individual subunits, protein subunits are a little bigger in eukaryotes, essentially, and there are more of them. But let's take a look at structure of ribosome in eukaryotes. So I am going to diagram this thing right here. Let's see how it comes out. Not too bad. So this is going to be the small subunit. So ribosomes have a large subunit and a small subunit. And this right here will be the large subunit. Okay, large subunit and small subunit. And if we stick these together, we are going to get an E site, a P site, and an A site. Okay, so E is going to be the exit site, and we'll see what that means in a moment. So again, this is the large subunit. Now, the small subunit in eukaryotes has a size of 40S, and S is for Svedbergs. So S is the Svedberg unit, and it's a measure of I guess rate of sedimentation. Rate of sedimentation. If you try to separate the components of a ribosome by centrifugation in a solution, I guess the rate of sedimentation of the small subunit is 40S. And interesting story. Somewhat interesting. My lab actually collaborates with a scientist who is the, I think, great nephew or great nephew of the scientist last name Svedberg, who the Svedberg unit is named after. So the large subunit has a size of 60S in eukaryotes. Now the small subunit is made up of proteins and RNA molecules. So the RNA molecule in the small subunit is called the 18S ribosomal RNA. In addition to the 18S ribosomal RNA, there are 33 proteins. So that's a lot of proteins in the small subunit. 33 different proteins in the 18S ribosomal RNA make up the small subunit. Now the large subunit contains 3 ribosomal RNAs, the 28S ribosomal RNA, the 5S ribosomal RNA, and the 5.8S ribosomal RNA. Now, in addition to these 3 ribosomal RNAs, there are 46 proteins in the large subunit. So we have altogether in the ribosome, if you can consider the small subunit and the large subunit, there are 1, 2, 3, 4 ribosomal RNAs. So the ribosomal RNAs, this is one of our examples of a non-coding RNA. Transfer RNA is another example of a non-coding RNA. And if you watch the video on exon splicing, we talked about the spliceosome. So there are snRNAs in the spliceosome. So those are small nuclear RNAs. And there are many, many, many more types of non-coding RNAs that we might get to when we in our section on epigenetics, which comes later in the semester. So where do these ribosomal RNAs come from? So let's briefly talk about the ribosomal RNAs. So, like we already talked about, in eukaryotes we have the 18S, the 5.8S, the 28S, and the 5S ribosomal RNAs. Now these are all transcribed by RNA polymerase 1. This one right here, transcribed by RNA polymerase 3. So what do the genes look like for these? Well, it turns out humans have hundreds of repeats of these ribosomal RNA genes. And what's the best way to explain this? So these three are encoded by a single gene. Of course, that gene is repeated hundreds of times. More on that in a moment. Let me diagram what this gene looks like. So let's say this is one rDNA or rRNA gene. So this part right here is going to be called the ETS. So let's say this is the +1 site right here. So this is where RNA polymerase 1 starts transcribing. This thing, ETS, this is going to be the external transcribed spacer. So that's sort of a strange name, right? And we'll see why it's called the ETS in a moment. So this part right here, this is the part of the gene that encodes the 18S ribosomal RNA. Then we're going to have what is called ITS1 right here. This piece right here, I'll color it in. This is the 5.8S ribosomal RNA encoding sequence in here. We have ITS2, which is right here. And then this big piece at the end, this will be the coding region for the 28S ribosomal RNA. Let's say the transcriptional cleavage site is right here. So what happens is RNA polymerase 1 is going to bind, let's say right here. We're going to call this the NTS. I'll explain why in a moment. But binds right around here, transcribes this whole gene. We get a long RNA molecule, and the 18S, the 5.8S, and the 28S are cut or cleaved from the long RNA to give us each of these individual RNA molecules. Now, ITS1, what does that stand for? That's internal transcribed spacer 1. ITS2, you can probably guess what that is, right? Internal transcribed spacer 2. So why are these internal? Because they're between the coding regions for the 18S and the 28S. Why is this the external? Because it's outside the coding regions for the RNA molecules. Notice these are transcribed, external transcribed and internal transcribed. This NTS, this is non-transcribed spacer. So in the human genome, now humans have somewhere between 200 and 600, and I think scientists are trying to figure out why some humans have way more copies of the ribosomal RNA genes than others. There's an interesting study, I think that came out last year, that shows that there's a huge variation between individuals. Scientists don't know why. And you know what? It's really hard to determine the number of copies in an individual human. So this large variation might be due to our inability to accurately measure the number of ribosomal RNA gene repeats in humans. But I'm sure in the next couple of years we'll learn more about copy number in humans and what it means for, say, human fitness, human health. So where are these found? So one thing I want to point out is that these are tandemly arranged in the human genome. So if this is at the NTS, and this is let's say this is a whole unit right here. Now this whole unit will be repeated many, many times on a chromosome, one after another. So here's the NTS of the next copy, and then here's the next transcriptional unit. Just copy this all the way over here on the other side here, and then repeat it many, many dozens of times. And I say dozens of times because these tandem arrays of ribosomal RNA genes are found in 5 different locations in the human genome. So there are 5 acrocentric chromosomes in humans. And maybe you remember from the first part of the course where we talked about what is an acrocentric chromosome. Well, it's a chromosome where one arm is way, way shorter than the other arm. And that way, way shorter arm is the P arm. And the long one's called the Q arm. Well, the P arms of these acrocentric chromosomes, which are 13, 14, chromosome number 13, 14, 15, 21, and 22. Well, the P arm contains each P arm contains dozens of repeats of the ribosomal RNA genes. It's pretty neat how they're found on the P arms and all the acrocentric chromosomes. Okay, so that's where 18S, 5.8S, and 28S ribosomal RNAs come from. So transcribed by RNA polymerase 1, they're cut from the one long RNA transcript. Sometimes transcripts like that are called polycistronic RNAs. Cistron is an old jargony term for genetics. Essentially, if there's multiple important things in an RNA molecule, we're calling that a polycistronic RNA. And they'll be cleaved from the RNA molecule by enzymes, and we're not really sure on the identities of those enzymes that do this processing, but the 18S, the 5.8S, and the 28S will be released, and then they can be used to make incorporated into the structure of the ribosomes. And you can imagine why we have many of these, right? Because we need lots of ribosomes to synthesize all the proteins our cells need. Now this one right here, these aren't the gene for the 5S ribosomal RNA. This is transcribed by RNA polymerase 3, and these are spread. I don't know how many copies there are. We can probably guess there are probably hundreds spread throughout the genome. So they're not arranged in tandem repeats like the ribosomal RNA gene for the other ribosomal RNAs. So just individual genes spread throughout the genome for the 5S ribosomal RNA gene. Okay, so now I think we're ready to talk about translation. So let's just briefly cover translation components. We have the small subunit and the large subunit, which we just went over. You know, if you're watching these videos, I wonder if you could watch it in double speed. I think you probably could. So, or you can try and, you know, hope some of your, you know, if you think this stuff is difficult, I hope you are taking notes, your own notes, as I am diagramming up these structures. So we need the small subunit and the large subunit. We need a messenger RNA, and we know in eukaryotes, the messenger RNA is going to look like this. Now, so at this part, you know, we focused on the structure of the eukaryotic ribosome. And in the notes, I guess the translation mechanism is focused on the way it works in E. coli. And, you know, here I'm pointing out eukaryotic specific modifications to the RNA. So for the most part, translation for I guess for what we are concerned with, you know, translation is going to work the same way in prokaryotes and eukaryotes. And we're not going to be too concerned with the slight differences between the mechanisms in both types of organisms. So initiation factors, essentially we're going to be talking, so in prokaryotes there are fewer initiation factors and elongation factors. There are more of those things in eukaryotes. The initiation process is a little different. You know, how the start codon is identified. We're not going to worry about those differences. We're not going to worry too much about the details of how the start codon is identified either. We are just going to try to remember how synthesis works, how translation works in general. So here I have 3 initiation factors, IF1, IF2, IF3. So these are prokaryotic initiation factors, but similar in eukaryotes. Elongation factors, EF2, TU, and EFG, and release factors. RF1, RF2. And then, I'm running out of room here, but amino, acyl, tRNA. Does everyone remember what an amino acyl tRNA is? So essentially that's another term for charged tRNA. So a tRNA that has an amino acid attached to the 3' end, sorry. So here is tRNA, 5' end phosphate, 3' end amino acid there. Here's our codon down there, anticodon. Okay, so those are all the components we're going to need. So, still recording here? Yes, we are. So, initiation. So, essentially, initiation factors plus ribosome assemble on messenger RNA and identify start codon. Yeah, that's it, really. So let me diagram that. Again, we're not going to worry about how the start codon is identified. In eukaryotes, it's supposed to involve some sort of scanning process. In eukaryotes, the ribosomes kind of assemble over the start codon. So let's say there's our start codon. Let's say the next codon is CGU. Now let me assemble this. Small subunit right over here. Okay, I should have been an artist, right? Amazing, my instructor is such a good diagrammer of ribosomes. That's what you're all thinking. Okay, small subunit, large subunit. Now let me put the initiation factors in here. So for our purposes, we just, you know, initiation factors are helping initiate translation. IF1, IF2, IF3, and, you know, I don't really know exactly where they're binding in here. So they're just helping with the process. They're necessary to get this process started. We don't need to worry about what scientists think they actually do. Okay, so here is the tRNA. It pairs. So this is the 5' end of the messenger RNA, right? This is the 3' end down here. So 5' end of the anticodon to the 3' end. And I'm going to put the amino acid way out here because the diagram's getting kind of messy. Okay, so we have initiation. We have the first tRNA, matches the AUG start codon. I have amino acid here, but you probably know that there's going to be a methionine here. And technically, you know, as these amino acids are being joined together, they're going to come out, the chain's going to come out a little hole at the bottom of the large subunit. And that's pretty neat, but I'm not talented enough to diagram that interesting aspect of translation. Not talented enough to diagram that on the fly. Okay, so that's initiation. Now this is the P site, so we're assembling. Initiation is starting with a transfer RNA bound to the codon, the AUG codon, that's in the P site. This is the E site, this is the A site. Now during elongation, transfer RNAs, amino acyl transfer RNAs are going to enter the A site with the help of elongation factors. And then transfer RNAs are going to exit the ribosome through the E site, the exit site. So let me diagram that next. Elongation. AUG CGU GCA. So I added another codon there. Now, let me see, how do we want to do this? Okay, we want the E site over here because, ooh, okay, that's not very good. So there's our small subunit. I'm not going to diagram the large subunit anymore. You just know it's there. So UAC, there's the anticodon of the tRNA. My loop there is getting pretty small, one of the loops. There's the amino acid. Okay, so this is the P site, this is the A site, this is the E site. Now another transfer RNA is going to come in here with the help of the elongation factors. So say elongation factors are helping now. Let's pair this up, complementary base pairing, not worrying about the wobble base pairing rules, allowing other non-standard base pairing to occur. Just put a GCA in there. Okay. Okay, so once we have a tRNA in the A site, paired up, and it has a suitable pairing interaction with the codon here, meaning the tRNA is correct, what happens is this amino acid is transferred over to this one. So a peptide bond, this bond is cleaved here, and a peptide bond forms between this AA, is formed between this amino acid and this amino acid. Right, so essentially what you're going to end up with is, well, I'll diagram that next. So as this is happening, what's going to happen also is the messenger RNA is going to shift 3 spaces. So we're going to put this AUG in the E site, CGU in the P site, and GCA in the A site. Oh my God, he's going to diagram that. Yes, that's why these videos are so long, right? You could watch it in double speed. I think you probably should. So here's the AUG, CGU, GCA, so E site, P site, A site. Okay, now we have that codon here in the E site. And doesn't need to be hydrogen bonded anymore. It's exiting the ribosome. There's no amino acid on there anymore. So we have this other amino acid in here. Now this is where the peptide, the growing peptide chain is connected to this transfer RNA. Did I say amino acid? To this transfer RNA in the P site. So I'm going to put methionine, right? Because we knew the first one was methionine. So methionine connected to another amino acid, whatever CGU codes for. So we have a dipeptide here connected to the 3' end of this transfer RNA in the P site. Now what's going to come in here? So the next tRNA is going to come in here and the whole process is going to repeat. So once this tRNA is loaded, we have acceptable hydrogen bonding occurring between the codon and the anticodon, indicating that the tRNA in here is correct. What will happen is this AA right here will be shifted over to the amino acid that's here. The messenger RNA will shift and we'll have I guess a tripeptide, so 3 amino acids linked together in a chain. And that process will occur over and over and over again. So that is elongation. And the messenger RNA is going through the ribosome 3 bases at a time. You could also picture it as the ribosome moving along the messenger RNA 3 bases at a time. Either envisioning the process either way is fine. Okay, so that's going to happen over and over and over again until a stop codon is encountered. So now the last thing we need to talk about, this is termination. So when a stop codon is encountered. And we've already talked about how there are no transfer RNAs for stop codons. Okay, let's see. E site, E site, P site, A site. And again, I'm not diagramming the large subunit. I'm only diagramming the small subunit. So there's something in the exit site here. No amino acid on this tRNA. Okay, let's say here there was an AAG. Phosphates there. And let's say here is UGA, and this is one of the 3 stop codons. Okay, stop codons, UGA, UAA, and what is the other one? I'm blanking. UAG. Then it doesn't really matter what comes after this, because they're not going to be translated. We have a stop codon here. So here, this is where that amino acid chain is. And it can be really long at this stage. And at this point, it's probably starting to fold. There are proteins called chaperones that bind to the amino acid chain that is coming out of the ribosome that help it fold into the right 3-dimensional shape. And so, depending on how the protein is, I could put, thousands of amino acids here. Or maybe it's a short polypeptide chain. It's a small protein. So I'm just going to put these little dots saying, it goes on for a long time. So this is the amino acid chain. And so when a stop codon enters the A site, what happens is one of the release factors enters the A site. We'll call this RF1. So RF1 and RF2. Now RF1 and RF2 is another release factor. Now these, I think RF1 recognizes 2 of the stop codons, and RF2 recognizes 2 also. So they each recognize a unique stop codon, and they both recognize so one unique one and one shared one for each release factor. We don't need to worry too much about those details. We only need to worry about that knowing the release factors recognize the stop codons. And when a release factor enters the A site and notices, and there's a stop codon there, what happens is this is cleaved. The amino acid chain on the tRNA in the P site is cleaved, and the protein is released. And then the ribosome falls apart. So we could, should I do it? I'll diagram the large subunit here. Okay. So once the release factor is in the A site, it recognizes the stop codon, causes this amino acid chain to be released from the transfer RNA in the P site. Protein is released, and then the ribosome dissociates from the messenger RNA. Okay, and we have translation synthesis. So that's it for translation. I hope that all made sense. Again, all that stuff is in the notes, so these videos are essentially just in addition to the notes, and I'm just kind of walking you through the notes with these little videos. If you like the videos, please keep watching them. If you don't have time for the videos, you should be able to get all of this from the lecture notes. So